Hey guys, how you doing? So welcome to chapter 2. We're going to look at the ultrastructure of cells, and we're going to look at cellular structure and the different organelles that form up the cells. Okay, so let's jump into it. We're going to break this series into two parts. One half we're going to look at the kind of the first few organelles within the cell, and then the next half we're going to look at the last few and put it all together into one. Okay, so here we go. All right, chapter 2, ultrastructure of cells. Before we start, we're going to look at sort of understanding cells. We use certain tools for looking at it. Now, this is just to give you a nice little overview to understand what are some of the different tools that we use in science for that. The two ones, one you've used and one you probably have heard of, but haven't used before. The first one is called an optical microscope, using light to illuminate specimens. This includes your light microscopes, the one we have in our class, the phase contrast microscope, laser scanning, confocal, and the fluorescent microscopes. These are all different types of microscopes that use, in essence, light of some sort to transfer and to communicate sort of the image that we want to see. Okay, now the second type of microscopes are what we call electron microscopes, which uses electrons to illuminate those images. And there are two main types. We've got the scanning electron microscope and the transmission electron microscope. So here is an image of what an optical microscope looks like, and then here is an image of your scanning electron microscope, which is right there. Scans the image and sends it over to the computer, and then the transmission electron microscopes. Now, one of the key things about the scanning electron microscopes is that they use electrons, and so what you're getting is a much more detailed image of your particular specimen, and you get it right down to the cellular level. However, scanning electron microscopes, because it uses electrons and not light, you don't actually get to see in color. And so often what happens is these images are digitalized and then colored in to accurately represent whatever it is that they're trying to show. Okay. Now, in terms of the scale, I think I've shown you guys something similar to this before, but when we're talking about in meters, right down to about 100 micrometers, you can see things with the unaided eye. Once you get into that stage, you usually tend to use the light microscopes to be able to see. You might be able to get, right down to maybe about your small scaled, and then once you get beyond a certain point, you tend to use your electron microscopes to be able to see the rest right up to your nanometer kind of range. Okay, now just a bit of a side story. There was a professor named Stephen Hell from Germany who actually shared a Nobel Prize in chemistry because he was able to use a particular type of, or discover a particular type of microscope called super resolved fluorescence microscopy. Okay, it allowed really high clarity for tiny molecules, and you can actually see right down to the organelle level, seeing the pores of the nucleus right in this particular image graph right there. So the photograph there, and so it's quite an accurate right down to the minute details of a cell, which really kind of helps us understand cellular structure and in turn their function as well. Okay, so that was a bit of a side story, a bit of kind of a nod to him for that. Okay, so what is the ultrastructure of cells? Well, in eukaryotic cells, that is cells that contain membrane-bound organelles, these are your plant and your animal cells, and they're characterized by having these structures within the cells themselves, which helps them function. Okay, they include a bunch of different organelles: the nucleus, mitochondria, endoplasmic reticulum, Golgi Complex, and the lysosomes, and a few others as well. These organelles are held in place by a 3D network of fine protein filaments and microtubules, which is known as the cytoskeleton. So together, you can think of the cell as a functioning machinery that contains all of these different parts, and each of the different parts have a particular role to play. And we're actually going to go through each of the major ones and give you guys a breakdown of what their functions are within the cell, so that you can understand the cell in a more kind of holistic sense. Okay. Here's a diagram of the animal cell. Now, I've shown this right at the start, but here's a bit more a detailed diagram of the animal cell, a typical animal cell showing some of the different organelles. Now, you're not going to need to know all of them. However, we are going to give you guys a very kind of thorough look at most of them, and by the end of it, you should be able to recognize most of these structures, if not at least the major ones. Okay, some of the key ones that you might already be familiar with: the nucleus, the mitochondria, and the cell membrane, which we looked at in the last chapter. Okay, there's a few others that you'll get used to: the vesicles, the Golgi apparatus, the lysosomes, the endoplasmic reticulum, and a few others as well. Okay, here's a diagram of the plant cell. You can see there's a few differences between plant cell and animal cells. Plant cells can photosynthesize, so that's characterized by a particular organelle called the chloroplast, which is this green thing here, and that's what allows the cell to capture light and then create their own food. You'll also notice that the plant cell has an extra layer around the outside called the cell wall, and that's what is characteristic about plant cells from animal cells, that animal cells don't have a cell wall, and so plant cells having the cell wall makes them rigid, but also protects them and keeps them structured as well. Okay, but once again, similar characteristics: the nucleus, the mitochondria, etc., being very similar in the structure as well. Both are eukaryotic because this is an organelle and it's got a membrane around it that allows it to function as it does. This is an organelle. This is an organelle. These are all organelles which have particular functions, which are found in eukaryotic cells, but not in prokaryotic cells. Okay. All right. Let's dive into it. In this video, we're going to look at the nucleus, the mitochondria, and probably the endoplasmic reticulum and the Golgi complex all together. And then the next video, we'll look at the other set. Okay. Let's start with the nucleus, probably the main thing that you kind of see when you see a diagram of the cell. That's what it looks like in the cell. It's that big red blotch in the middle. And it is the defining feature of eukaryotic cells. All eukaryotic cells have a nucleus, and inside the nucleus contains your DNA and things like that. Okay, so it's spherical, and it's enclosed in a double membrane called a nuclear envelope. Yeah. What the nuclear envelope contains is that it has these protein-lined channels called nuclear pore complexes, the NPCs, right? And so the NPCs are these kind of holes which separates the nucleus from the rest of the cell, okay? And it's what controls the exchange of materials between the nucleus and the cytoplasm, which is the rest of the cell, okay? So the NPCs are protein-lined channels and control the exchange of the materials, mainly looking at things like ribonucleic acid, like RNA, and then also proteins, including ribosomal proteins from within the nucleus to the rest of the cell. Yeah, so it is that kind of boundary. Think the cell membrane for the nucleus, okay? Now, inside the nucleus is also a second organelle called the nucleolus, or a sub-part of an organelle. The nucleolus is composed of ribonucleic acid, which is RNA. Now, this is slightly different from DNA, and I'll explain what the difference is in a second, but it produces ribosomal RNA, rRNA, which is really important for when cells need to produce their proteins. They use the RNA as a kind of a photocopy of the DNA. Okay, what are the functions of the nucleus? Well, the main function of the nucleus is that it controls DNA replication during cell division. Yeah, DNA is the blueprint of life, and so that genetic material needs to be replicated every time the cell divides, and so the nucleus controls that and manages that replication of genetic materials. It's where all of the DNA of the cell is stored and replicated whenever it's needed. Okay, it repairs the genetic material, so the DNA itself gets managed and repaired whenever it gets damaged and modified or mutated. And then it is also initiates the gene expression. Yeah. So what that means is it translates the genes into the proteins that the cell then uses to characterize whatever the organism is. Right. And it also controls metabolic activities of the cell by regulating which genes are expressed. Okay, so what that means is if you think about the nucleus as being the control center for the rest of the cell and then subsequently the rest of the organism, that's what the nucleus does. Okay, now most of you already know what DNA is, but let me just go through it again. So DNA stands for deoxyribonucleic acid. It is a type of biological molecule that is responsible for having or storing genetic information for an organism. Yeah. Now, my apologies that the image there kind of blocked the rest of the words, but yeah, it's genetic material and it stores biological information for the function of cells. Your cells are unique to you, and your DNA is unique to you, and no other organism or no other, member of your species will share the same DNA as you, unless you're identical twins. Okay, and so that storage of DNA allows you to express the features that you have. Your blue eyes, your brown hair, or your blonde hair are all characterized by your DNA. Okay, and the expression of that DNA is managed by the nucleus. Okay. Oh, sorry, let me go back there for 1 second. So here is the nucleus, and you can think of this as being your nuclear envelope on the outside there, and then these little holes is what allows the DNA to be photocopied and moved out in and out of the cell freely. Okay. Now, one of the key features, and we'll explain this a bit more later on when we look at proteins and things like that, is that the DNA actually doesn't leave the nucleus. It stays within the nucleus, and what actually happens is it gets what we call kind of like transcribed or photocopied by the nucleolus into an RNA strand, which is a single strand, and that RNA gets sent to the rest of the cell. So if you think about the nucleus as being a safe, it stores the blueprint of all the information of the individual, every single feature of the individual stored in there, rather than taking it in and out all the time from the nucleus. It gets photocopied, and those photocopy is what is being used as instruction for the rest of the cell. That way the DNA doesn't get damaged, doesn't get moved around the cell, but it stays within the nucleus and stored within the nucleus the whole time. Okay. So DNA exists inside the nucleus in the form of chromatins. Yeah, so that's the structure of the DNA. DNA is this double helix strand, but when it gets wrapped up and condensed, it forms these little kind of circular things called chromatins, and then they too get folded together, and eventually it folds together to form a chromosome. Yeah, and so during cell reproduction, the chromatins form the chromosomes that you kind of see when we were looking at genetics and stuff like that. Okay. All cells contain the same set of DNA. Okay, your blood cells may have started with the same DNA. Like white blood cells will have the same DNA as your muscle cells, as your brain cells, and things like that. However, in different cells, different genes are expressed. Some things are switched off. Yeah, your muscle genes are not going to use the nerve cell component of the genes, and so they get switched off, and only the muscle parts are being used. Yeah, and so that's how your body has all of the same DNA, but only some of it gets expressed in different types of cells. Yeah, your bone cells will not need the expression of eye color, for example. Most cells only have one nucleus, but some will have more than one. So for example, here's a diagram of a liver cell, and you can see that liver cells have two nuclei, which is the plural for nucleus. And some will have none. Blood cells, for example, will start off with a nucleus, but they actually lose the nucleus because they are kind of just transporting oxygen around the body, and so they don't need a nucleus. They're kind of like a transport vessel for the body. And some will have many. So, for example, muscle cells tend to have multiple nuclei, as a result of that. Yeah. Now, the next organelle that we're going to look at is the mitochondria. Now, the mitochondria is, in your diagram, this little brown-looking thing here with this kind of squiggly line running through it. Okay. Now, the mitochondria is the energy supply of the cells. It is the organelle responsible production of adenosine triphosphate. We looked at adenosine triphosphate in the first chapter, shortened as ATP. ATP is the energy currency of cells, and therefore the energy currency of all life. Yeah, your body uses ATP to be able to do the chemical reactions it does, which allows you to do the things you do. When you think, you use up ATP. When you run, do exercise, and you use, you work out and things like that, you also use ATP. When you eat food and your body needs to digest the food, it uses ATP. Anything that uses energy uses ATP, and the ATP is produced in the mitochondria. Okay, living things use energy all the time, and so the mitochondria is really important in supplying the cell with the ATP it needs so that it can do the activities that it does. Okay, now here is the chemical structure of adenosine triphosphate. Yeah, the adenosine is this component here. It is an adenine molecule together with a sugar base, or kind of like a sugar base, and then the triphosphate molecule sits at the back. There's a phosphate, there's a second phosphate, and there's a third phosphate, making a triphosphate molecules. We're going to explain how this ATP molecule works later. But that's the structure. That's what the mitochondria looks like, and it contains a few different components. And in the next chapter, in chapter 3, we're actually going to look at how energy production occurs in the mitochondria. And here is an electron microscope image of the mitochondria. So you can see there, that's the mitochondria there, and those little squiggly lines are those components within the mitochondria. Okay. The formula for cellular respiration, right, is the chemical reaction where glucose and oxygen gets converted into carbon dioxide and water, and in that process energy is being produced. Yeah, so here is a shortened version where glucose C6H12O6 plus 6O2, 6 molecules of oxygen, produces or gets converted into 6 molecules of carbon dioxide, 6 molecules of water, and a whole stack of ATP in the process. Okay. Here's the worded formula, and here is the chemical formula there. Okay. Now, the number of mitochondria in a cell will vary. In general, the more active the cell is, in other words, the more energy it requires, the more mitochondria it will have. Yeah, so here's a bit of a contrast. Yeah, for example, liver cells play a really important role in the breakdown of toxins in the body, and so they need a lot of energy in order to break down all the toxins that you get in your body. And so liver cells will generally have about 2,000 or so per cell, whereas mature blood cells will have none, because the blood cells themselves don't need energy. They just transport oxygen around the body. Okay. Muscle cells and things like that will have a few other amounts as well, up to about 50 or so mitochondria, I think, around there. Okay. Now, let's move on to the next one. So we're looking at ribosomes. Now, ribosomes are your protein factories. Okay. And so this organelle is responsible for the production of protein in your body. Now, if you don't know what proteins are, everything that your body is made up of is essentially protein. Yeah, your protein forms your muscles, it forms your muscle tissues, your brain fiber, your bones. Everything is pretty much made up of proteins, and so it is the structural component within your body. And so this protein is produced by your body from building blocks called amino acids. Yeah, so the amino acids, these protein building blocks, get assembled together to form the proteins, and then your cell then uses that for whatever it needs to use it for. Okay. And so all of that happens through this particular organelle called the ribosome. Okay, the ribosome is made up of 2/3 of RNA and then 1/3 of protein itself, and they are often very small, about 0.03 micrometers in size. They're kind of like these little small dots that are littered throughout the entire cell. And you can't see them with the naked eye or a light microscope. You can only see them with an electron microscope. Yeah, and often they are present in large numbers in a cell, because more often than not, cells are required to produce proteins in one shape or another, or one form of protein or another. And so there are many ribosomes found in cells, and especially in the ones that are required to produce large amounts of protein in the process. Okay. Oh, sorry, that shouldn't have happened. Now, there are kind of two locations where ribosomes can be found. The first one are what we call free floating. These free floating ones produce proteins that the cells themselves use within the cell itself. Okay, so the free floating ribosomes are what the cells will produce the proteins and then use the ones that they have produced. There are a second set of ribosomes that are found on another organelle called the endoplasmic reticulum, or the ER for short. And those are the ones where the cell produces in kind of like in factory mode, and then it exports it out of the cell and transport it around the organism to be used elsewhere. Okay, and so depending on where the ribosomes are found, that's what determines what's being used. Now, this little GIF that I've put in here sort of shows how the process is made. Yeah. I'm going to go look at this very briefly later on in the term in Unit 2. You're actually going to look at this in a kind of bit more specific detail around how the genetics play a role in it. But for now, this is sort of how it works, okay? So the ribosome is this little purple part here, yeah? It is made up of 2/3 protein, 2/3 RNA and 1/3 protein, and contains two major components. Here is the major subunit, and then here's the minor subunit. And what actually happens is, see this code that runs through the minor subunit like a conveyor belt? That is the RNA that is the photocopy of the DNA from the nucleus. And so the DNA in the nucleus gets photocopied or transcribed into an RNA. That RNA leaves the nucleus, goes to the ribosome, and sits on this conveyor belt that travels across. Now, depending on the code, what happens is these molecules called transfer RNA will go and collect amino acids and assemble them according to this code. And so the code matches the opposites of each of the nucleotides or the letters. And then they will collect a specific amino acid that matches these codes. And what happens is if it joins these amino acids together, and the building blocks then form your protein chain or polypeptide chain, and then that gets folded into the protein shape that needs to then be used by the body. And so that's how the ribosome works. It's like this conveyor belt factory work that assembles the building blocks together to form your protein, and then that protein either gets used by the cell or gets exported out of the cell and used elsewhere in the body. Okay. So that brings us to the endoplasmic reticulum, or the ER. The ER is a bunch of folded interconnected system of flattened channels, and they're responsible for transporting materials within the cells. Okay, there is the kind of the diagram of it, and if you looked at the cell inside a microscope image, often these kind of funny looking folds and layers in it is the ER. Now the ER is divided into two components. Okay, you've got what we call rough ER and then smooth ER, and I'm going to explain the functions of both. Okay, now rough ER is called rough ER because the texture of it is rough because it contains ribosomes on the outer surface. Yeah, these little dots here are the ribosomes that is attached to the ER. So this diagram shows rough ER, and so rough ER, because it contains those ribosomes, what it does is it's responsible for that folding of the protein or the polypeptide chain into the correct shape. Yeah, so once it gets assembled, it goes to the ER. The ER then folds it into the correct shape, and then it gets exported to the next organelle. And so it assembles the complex proteins such as hemoglobin and things like that. And also, it is also responsible for then attaching sugar groups to form glycoproteins, which is another set of biological molecules that your body uses. Now, if you have a good memory, you might remember glycoprotein appearing in chapter 1 when we looked at the glycoprotein markers on the plasma membrane, which forms the antigens or the signals of the cells, which helps the immune system identify the cells. That is one set of glycoprotein that your body uses. And so the ER is responsible for creating these things by folding them into the shape that they need to be, and then moving them to a different organelle for exporting. Okay, so rough ER, responsible for producing or folding proteins and assembling complex proteins and glycoproteins. Okay. The smooth ER, on the other hand, lacks those ribosomes, and so smooth ER is endoplasmic reticulum that does not have ribosomes, and because it doesn't have ribosomes, it's nothing to do with proteins. Okay, and so here's a diagram of the rough ER. So if you say that's the nucleus, there's the rough ER, and then by extension the smooth ER sits on the other side there. And what the smooth ER does is it's not responsible for protein production or assembling. It is responsible for the synthesis or the production of lipids. It produces lipids, which is a different class of biological molecules. Lipids include fats and oils and things like that in your body. And so your smooth ER is responsible for making these lipids. It is also responsible for creating hormones such as steroids in some particular glands. Okay, so in particular hormone producing glands, the smooth ER in those glands are also responsible for synthesizing steroids, hormones that you use, such as testosterone. In liver cells, the smooth ER plays a really important role in detoxifying drugs. Okay, certain drugs that go into your body needs to be detoxified, turned into a water-soluble form, so that your kidneys can then excrete them and get rid of them from your body. Okay, and so those liver cells will have, a higher proportion of smooth ER in order for them to detoxify those drugs. Okay, and also in liver cells, the smooth ER is then responsible for storing glycogen and then breaking it down into glucose whenever necessary. And so these glycogen molecules get stored by the smooth ER, and then it gets rebroken down into glucose whenever the cell needs to use them. Okay. Now this brings us to the last one of this particular video, which is the Golgi Complex, or otherwise known as the Golgi apparatus. Both words are synonymous and they mean the same thing or are referring to the same organelle. But the Golgi Complex is the last part in protein production. What it does is it then exports substances out of the cell or receives substances into the cell. Okay, so think of it as a delivery system. The Golgi complex also looks like a bunch of folds. However, the Golgi complex produces these vesicles, which then secretes and removes it out of the cell. So it exports substances out of the cell. All of the protein that gets produced by the ribosomes and then folded by the ER or the rough ER, then gets shifted into the Golgi complex through a transition vesicle, and then that gets then exported outside of the cell through the secretory vesicle. Okay, now a vesicle just means a small package. Okay, so a good example, pepsin, which is a digestive enzyme that is used for breaking down proteins. Okay, so for you to eat meat, you need pepsin because it breaks down the peptide bonds between a lot of your protein. Your DNA will have the genetic information to produce this enzyme pepsin. It gets transcribed into the RNA molecule sent to the ribosome. The ribosome then assembles it together into a molecule, sends it to the endoplasmic reticulum, the rough ER, which then folds it into the correct shape. And then what happens is the rough ER puts it into a transition vesicle, sends it to the Golgi complex, and then the Golgi complex will then put it into a secretory vesicle, send it out of the cell, and then through the bloodstream, and then into the location where it's necessary, most likely a location somewhere in the digestive system where it gets secreted into the stomach to break down protein. Okay. Other things include hormones, chemical signals, insulin, which regulates the sugar levels in your blood, and things like antibodies, which allows you to fight infection. All of these things are produced within the cell, but then not kept within the cell, but exported to other locations where it is necessary for it to function. And so that's what the Golgi complex does. It mails it out, sends it off, receives any new ones, and then sends it around the cell that is used. Okay. Here is what a diagram of it looks like. It is separated from the nucleus and it looks like a bunch of different folds like that, kind of similar to the endoplasmic reticulum, but not as kind of widespread. Okay. When exporting, the excretory vesicles move into the plasma membrane and they actually merge to discharge the contents. And that's the same process as exocytosis. So when we're talking about exocytosis in chapter 1, that's what we're talking about. The Golgi complex is responsible for those vesicles, which sends it out, attaches to the plasma membrane, and then releases the fold and the contents into the bloodstream, which then gets transported around the body. Okay, so we're going to stop it there for this video. Hopefully you guys have learned something new there, and I'll catch you guys in the next video.